Today I will explain you about Cornybacterium diphtheriae and the bacteria causing upper respiratory tract infection. The bacteria that cause upper respiratory tract infections are Streptococcus pneumoniae, Haemophilus influenza, Staphylococcus aureus and Streptococcus pyogenes. These are the bacteria that cause the upper respiratory tract infection. Now, moving to the morphology of Cornybacterium diphtheria, it causes diphtheria. They are thin, slender, gram-positive bacilli with pleomorphic character with them. They have metachromatic granules on one or both the ends. These are the metachromatic granules which are present in one or both the ends. It takes the stain Leophrys methylene blue stain. Granules are stained up with a bluish purple color. Hence, they are called as metachromatic granules. They are arranged in an angular fashion of V shape or L shape. Hence, this is also called as Chinese letter or cuneiform arrangement. This cuneiform arrangement is due to the incomplete separation of the daughter cells by binary fission. Now, moving on to the pathogenesis. First, the bacilli enters by inhalation. Later, there is lodgement and multiplication. Exotoxin production takes place. Local tissue necrosis. Toxin absorption. From here, local tissue necrosis is divided into two parts. One is toxic absorption and multiplication of exotoxin product. This toxic absorption causes toxemia, which in turn causes affinity for myocardium, nerve endings, adrenal gland. Now, when it divides into multiplication exotoxin production, that causes formation of diphtheria pseudomembrane. That in turn causes mechanical obstruction. Next, let's move on to the lab diagnosis. Lab diagnosis, first we have to do isolation of organisms. One is specimen, how you take up the specimen for the test. One is throat swab or portion of the membrane is taken, the infected membrane. Direct smear, gram stain and Albert stain. Gram stain, it shows club-shaped gram-positive bacilli Chinese letter, the one which we saw here. That is gram-stained. Albert stain, it is green bacilli with metachromatic granules. You have seen on top there. Culture media, it shows actively participation in culture media like enriched and selective. In enriched media, we can use blood agar, chocolate agar, Leophrys serum sloop. Selective media, we can use telluride blood agar, that is black color colonies it produces. Biochemical identification, his serum sugar media and it is urease negative. Diphtheria toxin demonstration in vivo tests. Lastly, the in vitro test contains ELIX gel precipitation test, PCR and ELSA. This was all about Cornybacterium diphtheriae. For further videos, please subscribe to our channel and press the bell icon.